I'm excited to share my experience, both as an addict and also as a neuroscientist. I should cut to the chase. I started using, as Lonnie said, at 13, and for the next 10 years, I took as much of anything I could find as I could. I was one of those who clearly had a predisposition. And when I learned that I had the disease of addiction, I thought, well, diseases, they can be cured, right? So I figured in my sort of deluded state that I would fix the disease and I'd be able to use. So that was my goal. And even though I had been kicked out of 3 schools at that time, the first one in 9th grade and then 2 colleges, I was determined, and that goes a long way. And so I ended up eventually graduating from college and then going to graduate school and then doing a postdoc, and I've spent the last almost 30 years researching the biological basis of addiction. So my goal is to summarize all of that for you tonight so that even though we haven't solved it, and I sure haven't solved it, there is quite a lot we understand about the neurobiology of drug addiction and drug abuse, and so that's what I'm trying to give you a snapshot of. So thank you for being here. The first thing I want to point out is that we are set up to use drugs that change the way we think and feel and behave. This is a really ancient tendency in humans, for sure, since the beginning of recorded time and even before recorded time. We find evidence of pot shards that have fermented drinks in them, and it just seems, and paintings on the walls of caves that show kind of psychedelic pictures with the mushrooms right next to them. So this is not new, and it's almost universal. I say almost universal because there are a few communities, mostly in the Inuit communities in the Arctic, that have the misfortune of not being able to grow anything, and so there are apparently not a single mind-altering chemical to be found in a few places on Earth. And in those places, people haven't been able to use them. And not only humans, but throughout the animal kingdom, people use drugs. So, I mean, other animals use drugs. Birds look for fermented berries sometimes and purposely seem to take several of those until they're kind of tipsy. Elephants are known periodically to break down distilleries, or to, I guess it's hard to stop a herd of elephants, so they come right in, drink it up, and then everybody tries to get out of their way, but they do this on a kind of a semi-regular basis. So the birds, the mammals, my favorite example is from insects. I don't know why this is so amazing to me, but there's a species of ant in South America that takes the time and effort in its ant mound to raise this certain kind of beetle. And these beetles are apparently very special because on the little beetle hairs on the back of their leg, they grow a fungus that the ants are interested in. And so the ants raise the beetles, they take care of them, they feed them, and this means that they can't have so many ant babies that they feed and take care of, so they sort of cut out some of their own interests to raise these beetles, and every once in a while they harvest the fungus. They all together eat it, and then they get very, very slow for like a day and a half. And there's apparently no nutritional value, but this is just a drive. Some people have argued, just like we have a drive for thirst and a drive for food and a drive for sex, we have a drive to kind of mess with our minds. And I think some of us maybe appreciate that more than others, but it's widespread. And in a short way, I want to tell you that what all these animal species have in common is a pathway that we've called the reward pathway, and it involves the neurotransmitter dopamine. And that dopamine pathway is one that evolved so that we would participate and find things reinforcing or pleasurable that would forward our survival. So what we find pleasurable, food and sex, are pleasurable to us because they help us survive. They help our progeny survive. If we didn't eat and we didn't procreate, that would be the end. So we find these things pleasurable, and they help our survival, and all abused drugs co-opt that same system. So plants and animals, fungi and things, made chemicals that co-opted this same system so that those plants could survive. So it's really an interesting kind of way that we evolved. So first I just wanted a little terminology out of the way. So a drug is any substance that's not food. And you probably might wonder, well, then what about chocolate or hot sauce? Are those food or are those drugs? Actually, rats will self-administer hot sauce. They'll press a bar to get hot sauce delivery because it is kind of more like a drug, and chocolate also. But anyway, a substance other than food that alters bodily function, and we're particularly interested in psychoactive drugs, and so those are drugs other than food that alter our perceptions, our moods, and the way we think and feel. What about other things like gambling or sex addictions or exercise addictions or shopping or Facebooking or all kinds of things? Are these addictive too? And the answer is they can be, because they also co-opt that dopamine system. So going back to the dopamine system, the theory that's kind of central to all research on drug abuse was developed in the 1970s by a guy, Roy Wise, and he argued that the rewarding value of any reinforcer, gambling or sex or cocaine, is related to the dopamine activity in this Mesolimbic pathway. So, and one of the things that comes from that is that our differences in that pathway are part of what is different about our risk for addiction. So it's very clear from tons of research over the last 50 years that some people have a very sensitive dopamine pathway that is sort of hungry for dopamine more than others, and others is maybe less sensitive, and that influences your risk. So just to kind of show a picture of that so you can put something with it, it's a really small set of neurons. It's not a very large pathway. It goes from the midbrain, which is kind of at the top of the brainstem there, so right here, to the limbic system, and that's why it's called Mesolimbic, because mid is meso. And this little pathway, the cells or the neurons here squirt out dopamine here, and anytime that happens, it feels good. And it's not good like a good mood, it's good like foreplay or chocolate cake or a shot of a drug. So it's a kind of a titillating, wonderful feeling of goodness. And we could obliterate the risk for addiction if we just cut out everybody's mesolimbic pathway. This has been tried actually, and apparently is still a little bit of a treatment in countries other than ours, a few other countries where they're just decide, well, if we block the ability to experience any reward, then people won't deliver drugs. And that's true. Cocaine would have no effect, marijuana, nicotine would have no rewarding effect. The problem is neither would other things like great music or a wonderful idea or falling in love, so all of those would be diminished too. But this evolved through natural selection, and it promotes eating and reproduction. It's co-opted by all drugs of abuse, and obviously drugs are often more potent than natural reinforcers. And there's a couple of reasons for that, but one is that we control the dose and the delivery. So even if your date is really a wonderful person, it's not quite the same as a bag of drug that you can deliver kind of on demand. And the other point about this is when we use these very potent reinforcers, we dampen the natural system. So it kind of like listening to very loud music will make you less sensitive to sound. Driving this dopamine pathway really hard, really frequently, makes the whole thing downregulate or become less sensitive, which of course then promotes more use, because one way to experience reinforcement or reward or pleasure is to turn the system back on. Okay, so I'm going to just summarize 3 main laws of pharmacology, and then we'll talk about some specific drugs. The first one, this is true about all drugs, they only alter the rate of what's already going on. And that's kind of obvious in what we just talked about, because dopamine is activating this, drugs are activating this Mesolimbic pathway. So in this case, drugs of abuse, among other things, are increasing the activity in this natural Mesolimbic pathway. But that's always true. Drugs can't do anything new to the brain because they wouldn't, they have to act on the substrates that are already there. We'll talk about that some more. Every single drug has a side effect, and the reason for that is that drugs acting on these particular substrates, like the Mesolimbic pathway and other pathways, in the case of the Mesolimbic, for instance, dopamine is there, but dopamine is also in other areas, like areas that mediate movement and cognition. So all drugs that influence dopamine could influence pleasure and movement and cognition. Serotonin, we know of probably selective serotonin reuptake inhibitors for depression. Serotonin is involved not just in mood, like depression, but also in sleeping and in eating and in sexual behavior. So the side effects of SSRIs go to sleeping and eating and sex as well. So it's true that they all have side effects. And then the most important law or axiom is that, and the one I'm going to spend most time on, is that whatever a drug does that affects the brain, so if a drug is acting on the brain to change our thinking or our feeling or our behavior, the brain counteracts that drug. And it does that to maintain homeostasis. And that, especially that last point, leads to kind of the core of addiction. So addiction is characterized by an overwhelming desire to use. This is a compulsion or an obsession. My first good drink, as Lonnie says, I had little sips of things at parties and stuff with my family, I guess. But when I was 13, I was staying over at a friend's house, and we went into her parents' basement, and we drank a gallon of wine together. And I'm pretty sure, because this is kind of my MO, but I'm pretty sure I got more than half of that. And I have to say, it was such an amazing, like a transcendent experience. I felt like complete. I felt well. I felt whole. I felt like enough. And it was really like a deep breath. I was like, aha. And up to that point, I had normal teenage angst, I guess. I was maybe a little insecure and a little worried about things and maybe a little toward depression, I don't know. But I drank that alcohol and I thought I had the ticket to living as an adult maybe, and certainly to negotiating my adolescence. And as I said, that experience, which I never quite replicated in terms of wonderful feeling of oceanic, wholeness, I did try to seek over and over and over again. And what I'm going to describe to you, both from a neurobiological perspective and my own perspective, is that the more I sought it, the more it diminished, the harder it was to find. Until by the time I was just turned 23, I was more insecure, more depressed, more anxious, and more kind of lost than I had been when I started. So anyway, there is this compulsion, there's a tendency to increase the dose, and there is dependence. And dependence is characterized by withdrawal when you take away the drug. And this has been probably the most contentious of the three, of these three. So in fact, when I hit bottom in 1986, and as I said, I was young, and what drove it, I think, so fast was cocaine, which was really prevalent. I lived in South Florida then, and so cocaine was the big thing in the 80s, kind of like opiates are the big thing now. And it's such a brutal drug that it kind of precipitated a fast bottom. But at the shortly before this, so it was in the 80s that people finally realized cocaine was addictive. They thought it wasn't addictive. It wasn't even an addictive drug because there was no physical withdrawal. They were using examples like alcohol and opiates, where you have DTs or shakes and real clear signs of physical withdrawal. And with cocaine, you don't feel like that at all. You just feel like there's nothing to live for because the world is so depressing. So finally, we recognize that there's a psychological tolerance or psychological dependence. The drug has to be bad for you and for society. So I love caffeine, but I don't think I'm addicted because there's nothing wrong with caffeine. And then, of course, the last one is the denial of the drug problem. So it may just be that I'm in denial about my caffeine habit. Okay, so these, I notice I have I was going to say, I don't know why I have another 80 years here. I apologize for that. But I want to, anyway, let me tell you what I was going to say about this. So why we have addiction is because the brain adapts to any drug that it gets regularly. And that adaptation is an attempt to maintain homeostasis. And homeostasis is a concept that came from physiology. So it came from physiologists in the 19th century, and then was understood even better by 20th century physiologists who recognized that there was kind of a balance in our physical states. So body temperature, for instance, is maintained at a stable level, about 98.6. If we get too cold, we shiver, which is purposely to generate heat. If we get too hot, we sweat, which is to dissipate heat. So in both cases, we're kind of maintaining this stable state, 98.6, through change, through making heat or taking away heat. And so another example of this, and actually Walter Cannon also, in addition to coining the term homeostasis, he coined the term fight or flight, which everybody has heard of. And he was kind of studying the homeostatic response to a stressful stimulus. And you know that if you're exposed to a challenge, you engage your autonomic nervous system, your sort of involuntary nervous system, to arousal, and you get into this fight or flight state, as you would maybe if you were taking a tough exam. So this is something that probably many of us can relate to, being in a stressful situation like a test. And once that exam is over, what Cannon noticed was that you don't just go right back to normal life. What happens after a big stressful exam or a talk? So this person's probably not tired or hungry here, but then right after the exam, something like this, probably after a big bowl of cereal or something. So we go from kind of one extreme to the other in a balanced way. And I have a personal story about that where I kind of understood this even better, just to illustrate the point. I was at Mesa Verde National Park in southwest Colorado, and it was early in the morning, and there was an RV. I was clean and sober, that's why I was able to be out at the parks, but anyway, early in the morning. Anyway, I was following an RV down the road, and it went right over a squirrel. And the squirrel, was way below the RV, so it was fine. It was just standing in the road when I came up to it in my little car. So I was thinking, well, get out of the road. Just stood there. And so I pulled my car off to the side, and I walked up to the squirrel, and I was like, come on. It wasn't bleeding or anything. I could see its little heart going like this. And so I get about 2 feet away, and I guess I thought I would go like this, and it keeled over dead. I know. And I was in grad school at the time, so I thought, what the heck? How did it die of fright? Nothing hurt it. And it died of fright. And the way you die of fright, actually, is not usually by the fight or flight, by the big arousal. It's by the counterposing response, which is to calm you down. And he was so aroused that he calmed down so much that he stopped his heart. Yeah. And that's called parasympathetic overshoot. And some people take advantage of that parasympathetic overshoot, like people who skydive. They don't skydive because they love the really stressful experience of thinking you're going to splat onto the ground. They skydive because when they land and they're still alive, they get the opposite response. So the point is that our physiology, our body responses, are kind of in this balance. And it's not that we're aroused and then back to normal. It's that we're aroused and then sedated or really relaxed. Okay. So this theory of homeostasis in physiology was adopted by two psychologists at the University of Pennsylvania in the 1970s who were studying psychological states or feeling states especially, so affect. And what they did is they took the idea of homeostasis and they applied it to feelings. And they said that any stimulus that changes our feeling state will elicit a response by the brain that is exactly the opposite to the effect of the stimulus. So there's really simple examples of that. If you're afraid, like you would be jumping out of an airplane, they predicted when the stimulus that caused that fear stops or ends, you would feel really happy. If you were, or relaxed, I guess I should say. This is a relaxed emoji, you probably knew that. I'm not so good at these. If you feel really happy and elated, great things are happening, you then are often let down after that. So you don't just go right back to your basic level. And I think there are lots of examples of this that you probably can imagine or maybe you've experienced. So if you find a lump that's not supposed to be there somewhere in your body, you'd be probably anxious and nervous and tense and worried for as long as it took to maybe get a biopsy done and then find out, oh, it's benign. And when that lump is benign, would you just go back to business as usual? Probably not. You probably have a few days of really being especially pleased with life and yourself and your situation, even though nothing, much had changed. Or if you're away on vacation, it's just a wonderful, great time. Does everybody experience this kind of letdown when you get home and it's like the same chores and the same house? Your house is kind of worse after a vacation than it was before, isn't it? And falling in love is a classic one. So when you're in the initial stages of falling in love, which is, by the way, just like cocaine on the brain, you're, full of, the world is bright and wonderful. You get used to your partner and it's maybe not quite so wonderful. Not terrible, but it's good, it's still good. And then something happens and you don't go back to your original state, you go to deep bereavement. So the time course for these things can be really long, but they argued that there is this kind of yin and yang or balancing act with feelings as well. And that's really relevant when it comes to drugs of abuse. So this is the pattern that they described for any stimulus, including drugs we'll see, that changes the way you feel. And what happens is if the stimulus comes on right here, this is time, comes on right here, you have this strong feeling. It could be the stimulus is jumping out of an airplane. And then the strong feeling would be, so it doesn't matter if it's good or bad, but it's a change from your neutral state, which peaks early, and then, and then adapts. I'm sorry, I'm pushing the wrong buttons here. And then kind of adapts, and then it goes below this. So if you were panicking at the airplane, then you feel relaxed. But with a psychoactive drug, if you feel a good euphoric feeling because dopamine is released, that kind of, you get used to that goes down a little bit, and then you always have this dip. And there is always a dip in dopamine and in other things after the drug. So to be more specific about that, they describe this pattern, which I just showed you, kind of looking like this, in terms of two processes. And this is really, even though they did this in the 70s, and you could read this paper yourselves, it's still the kind of central theme of addiction neurobiology. So many researchers around the world focus kind of on this sort of model. So they argued that the reason our experiences are like this is what we feel, the manifest affective responses, our feeling state, excuse me. We do that for two reasons. There's an A process, and that A process is the brain's response to the drug. So the drug does something to the brain. If it's cocaine, it blocks reuptake of dopamine, so there's more dopamine in the synapse. If it's an opioid or like a narcotic, heroin or fentanyl, it activates opioid receptors. So any drug, alcohol, does a bunch of things. And all of that, the drug effect on the brain produces this A process. And the brain produces a B process. The brain doesn't want to be off of your homeostatic baseline, the neutral state, so it counteracts the effect. And it makes its own compensation to it. And when you combine the A process from the drug and the B process from the brain, you get this manifest response up here. Okay, so what's interesting, especially interesting about that, is that the brain is so clever, it's really clever, it learns. If you take the same amount of drug over time, that drug produces the same A process, but the B process comes on earlier, is larger, and lasts longer because the brain adapts. And if it's nothing else, I mean, I'd say the brain is really two things. It senses contrast, it senses something's happened, and then it learns from that. Those are its main functions. So because of this growing B process, you can see how the same amount of drug produces less of an effect. That's tolerance. And you can see how when you get rid of the drug, it goes away, you have this big withdrawal. And that's dependence. And those are the two hallmarks of addiction, two of the five, really. So one example, just to sort of illustrate this kind of quickly, we'll go through more later, but is that the acute effects of any drug are always the exact opposite of the withdrawal effects, which may be obvious, but whatever you're taking a drug to do, in my case I take caffeine so I can wake up and hit the day running, that means that when I wake up and I haven't had a cup of coffee, I'm not running. I'm also not probably speaking in complete sentences. I'm really, really hardly awake. So I don't wake up until I get maybe a cup and a half in. And then I'm not, wildly awake, I just feel normal. So here you can see that opiates produce analgesia, which is to block pain. They reduce your respirations. That slows down your breathing, and that's actually how you die of an overdose. Your breathing stops. They produce euphoria. They help you relax and get some sleep, tranquil. They decrease your blood pressure, cause constipation. And in each case, the withdrawal sign is the exact opposite. And that's true for every drug. And the way we understand that is in terms of this pattern of affective dynamics. And what Solomon and Corbit argued is that it's so important to maintain this stable, neutral state that your brain counteracts any perturbation from that neutral state. And I think, why would that be so important? Why do we have to have a B process that's so, ruins all the fun, you could say? Well, if I depict my brain's activity as sort of like this, so this is what's going on in my brain, nothing much is happening, and then something happens, it would also cause a sort of a bunch of activity, and I'm just depicting it this way. It's kind of hard to tell, isn't it, that something happened? Whereas if my brain activity looked like this, was kind of stable and steady. And then something happened, these two are exactly the same size, I could really tell. So what I'm saying is that our ability to know if anything's going on, anything important happened, an opportunity for food or for, an interesting experience. If I want to know if anything happened, I have to have a stable baseline. And so in order to ensure that I do, the brain is able to compensate for any perturbation. And there's one more point about this. I said that the B process comes on earlier and lasts longer and is bigger, and those are responsible for tolerance and withdrawal. But this B process the brain is so clever that we don't even have to wait for the drug to come to elicit a B process. We can sort of learn to produce a B process like Pavlov's dogs learned to salivate to the bell. So you might remember Pavlov was studying learning, and every time the dogs got some food, he rang a bell. And so the dogs, as soon as they heard the bell, they would start to salivate without food there. And that's because they were anticipating that the food was coming. The bell predicted the food. Well, our brain does that with drugs too, so that we have lots of things that will elicit a B process. The drug will do it, of course, but also paraphernalia for the drug. So just seeing a baggie or just seeing a bar, if you drink at this bar, will elicit it. Or your friends who you use with. Or maybe it's the time of day. So some people have a drink every afternoon at 5:30. And if they don't get it at 5:30, they would get increasingly, what do you think? Since alcohol is a relaxant, it makes you calm, they get increasingly anxious and stressed and tense because their B process comes on as a result of the time. Emotional states are also able to elicit a B process. So being tense and anxious would precipitate wanting to pick up, because that is the B process. Money for some people. So if you're a Coke addict and you're trying, or any kind really, you're dependent on having some cash to score, and you get a wad of cash in your hand, then you'll probably start experiencing the B process, because that money predicts that you're about to get high. And so it could be anything. It could be weather, certain kind of weather. I think I still have this experience. If it's a kind of, I don't know if it's the humidity, I said I hit bottom in South Florida, but so if I hear, it's a kind of a warm breeze or something, and I just feel like a margarita. I haven't had a margarita in 30 years, but my mouth kind of puckers, because I think, wow, that sounds good, out of the blue. So these are cues that are able to elicit the B process itself. And what does this all translate to? It's that other sign of addiction. It's craving. So all of these things will elicit craving in an addict. And I think this is used in treatment centers now because we used to have the idea, look, if we can just get the addict away from their cues, their environmental cues, get them out of the situation they were in, away from their friends, away from their the areas or the neighborhoods that they picked up in, and take them out somewhere else. That's how they used to do it, and it would work okay until you brought them back to that context. And then out of the blue, one day they were feeling perfectly fine walking down the street. Maybe they bump into somebody they used to use with, or to their dealer they haven't seen in 2 years. And all of a sudden they're sweaty and anxious. So now instead we do things like expose users to a pipe with baking soda instead of cocaine, or to something that looks like alcohol but doesn't give it, or even to frustration, and see what happens, to help them not use during that time. Okay, so I want to talk about, this is sort of the general basis of this kind of the psychopharmacology of drug addiction. And now I want to just go through a couple of examples for the rest of my time. So one is alcohol, and this is wildly popular. It's been popular, as I said, since the beginning of time. Alcohol comes from fermented fruit, so it's produced naturally. Anybody can make it. Doesn't cost anything. You just need to let the fruit sit around a while. We now distill it, so it gets even stronger, but it's very popular. And about 8%, I said a small percentage. I guess it's small compared to 100, but about 12 or 13, 1 out of 8, actually, this is a mistake. It's 1 out of 8 people. It's about 12 or 13% of the adult population have a problem with alcohol. Okay, so it's in fact more problematic than all other abused drugs combined. So it's more lethal than opiates and kills more people than opiates or any other drug, except for tobacco. And it's increasing, especially in young people, although we did this when I was in college too, but binge drinking is as popular or more popular than ever. The binge drinking is having 4 drinks if you're a woman or 5 drinks if you're a man in about 2 hours, and that brings your blood alcohol level up past the legal limit, and that is particularly prevalent among young people, and that habit or that activity is really increasing the risk for alcohol use disorder and other addictions in those people. So one of the statistics about this is for every year before 21 that you start drinking alcohol, you increase your risk for having an alcohol use disorder by about 7%. So if you can wait to 21, you're not going to be in that 1 out of 8, much less likely. But if you start like I did at 13, then your risk goes up to about 50% at least. So part of the risk for developing an addiction is the age at which you begin using. And certainly that contributes a lot. In fact, 80% of people who have any substance use disorder begin using before they're 18. And I told my kids, I tell my students this too, I don't know how well it works, but I say if you can just hold off until you're 23 or 24, or maybe even 25, then you probably, it's much more likely you'll be able to use kind of with impunity as a social user. But if you have to start early, it's not so great. So what are the factors? One of them is using early, and others are things like your personality. So certain types, certain people are more at risk than others. People who are interested in novelty seeking, for instance, who are more impulsive, for instance, or who are not so swayed by punishments. Those people are psychologically at risk. There's also a lot of sociocultural factors, like how accessible the drugs are. If you happen to be hanging around in dorm rooms like I was, where everybody was binge drinking, then that's more likely. But also a lot of biological factors, and those are the ones that I mostly focused on. So all 3 of these combined, and this could be alcohol abuse or it could be any drug abuse. So the thing that I thought was going to be sort of quick to understand turned out not to be so quick. But I wanted to just share a little bit about this genetic risk. So we know that about half of your tendency to become pretty much any kind of substance abuser is due to genetics. We know that your tendency to have a problem runs in families, and we know we have the genome cloned, so we know all the DNA in human beings. Yet we really don't know most of the genes still. So part of the reason for that is that there seem to be maybe hundreds. There's definitely over 50 genes, and they're each contributing just a tiny amount. And actually, there's probably 50 to 100 genes increasing your risk, and probably 50 to 100 genes decreasing your risk. So these all combine in really complicated ways. But one of the genetic risks has to do with beta endorphin. And I think this kind of illustrates what's different about the neurobiology of those who are prone from those who are not. So beta endorphin is a peptide. It's a neurotransmitter in the brain, and it's one that is mimicked by morphine and heroin, so it's an opioid. It does the same things as morphine and heroin. It makes you feel relaxed, and it blocks pain, and it blocks stress, like that list I showed you earlier. And it turns out that beta endorphin levels differ based on your family history. So if you have no alcoholics in your family, this is like a negative sign here. So there's really in your parents and your grandparents have no alcohol problems. You start out with about twice as much beta endorphin in your blood and probably in your brain as people who have 2 or more alcoholics in their family. These people start out with much less. Interestingly, if we give them alcohol, as was done in this experiment, so we deliver low or medium or high doses of alcohol, for those with the negative family history, there's really not much change in beta endorphin. But look what happens if you have alcoholics in your family. And by the way, these subjects were children who hadn't really had a drink yet. Probably children like me, okay? So what happens is you give them a little bit of alcohol and beta endorphin levels start going up. And you completely sort of get rid of the deficiency in beta endorphin, and you can even kind of augment it. And I think that it's easy to imagine from this that when the people on the left experience alcohol and the people on the right experience alcohol, they have 2 different experiences. The ones on the left might feel pretty good to begin with, and then they drink and it's okay. Yeah, I might do that again. And the ones on the right might have an experience like I did, which was, oh my gosh, are you kidding? This is great. This is the key. So not only dopamine levels and variation in dopamine, but variation in endorphin and variation in many other peptides and compounds contribute. Now, I told you that the brain will adapt, and it does. So one of the B processes for alcohol, this is a bummer, and this probably happened to me, is that the brain stops making beta endorphin. In regular drinkers, there's very little or no beta endorphin. And that, of course, would give you the right-hand column of that opiate table. So they're more sensitive to pain, more sensitive to stress, more anxious, difficulty sleeping. You can understand. All right, I want to give you another example. I think we're doing okay on time, sort of. Close to my heart, too. So even though I loved alcohol, I really loved weed. It's funny, it was just the perfect drug for me. That's not true for everybody because of biological differences. So some people it makes paranoid and some people it makes anxious. For me, it made the world just interesting and lovely. So I want to tell you about the A process, and then I'll tell you about the B process. And the A process really starts with studies done in the 1990s when we discovered how marijuana was producing its effects. And so this is the first paper that showed this in 1991. And what they showed was that they found the place in the brain where marijuana was acting. And it was kind of a big surprise to everybody because the place in the brain where marijuana was acting was sort of everywhere. All the black dots on the board are the places where marijuana acted. And if I had time and I showed you all the places where cocaine acted, or even alcohol or nicotine, it would be very few spots. This was almost every synapse, almost every cell is capable of responding to marijuana. And the places that it responds are called CB1 receptors, and those are the same receptors that are activated by THC. So why would your brain make all these receptors for marijuana? It's not doing it in case you find these plants and smoke them. It's doing it because there's a natural compound that marijuana is mimicking. And in fact, there are two of them, and they were discovered a few years after the receptors. They're called anandamide and 2-AG, or 2-arachidonyl glycerol. And these are the endocannabinoids, the natural THC-like compounds. So THC only works because it speeds up, increases the rate of what's already going on. And so what already going on is this cannabinoid signaling. So what is that doing? Well, in the brain, there's neurons, as you know, and typically the neurons receive input around their soma or their cell body, and they conduct it electrically. This is sort of the code of the nervous system down their axons. And then the axons branch usually, and there's a little gap between the axons of one cell and the soma of the next cell. And that gap is the synapse, and 98% of drugs work by affecting the synapse. So we looked there for THC's effect, and to describe that, I kind of blew this up, the synapse up. So normally what happens is neurotransmitters are released from the end of one neuron to act across the synapse to the next cell. And that next cell has receptors like dopamine receptors or acetylcholine receptors or serotonin receptors. In the case of cannabinoids, the communication goes in the opposite direction. This was a really novel mechanism for people to try to understand. So here is the cell, it releases the neurotransmitter. Maybe that neurotransmitter is glutamate or GABA or another neurotransmitter. Those neurotransmitters interact on this cell across the synapse. And then these little C's are cannabinoid molecules. So anandamide or 2-AG that get released and go back and talk to the first cell. This was a really new mechanism, and as I said, it's in practically every synapse. So what does this mean? I have a picture of my dog to help explain that, my cute dog, because I think I can illustrate it good with him. So what this indicates is that some message comes across the synapse, and then the endocannabinoids are synthesized and released to go back and sort of send a message, sort of a bidirectional message across the synapse. And that, we think, acts like a highlighter to say the message that just came through was something really important, was something salient. And the reason it's all over the brain is because you never know which synapses are going to be transmitting important or critical information. So this helps us decide what to learn and how to learn, and it helps us sort important information from not important information. So Bowden, one day, my dog was out in the yard as a puppy. Actually, he was about this age, and my daughter dropped a piece of bacon in the grass. And you can bet that was important for the dog. That was like a big day. And so what I would say to illustrate this is that his brain, he found the bacon, and as he did, his olfactory system got turned on, his motor system got turned on, his learning and memory. Wow, this is great. What is it? And so anandamide probably got released in many structures, in many circuits, to tell Bowden, okay, something really great just happened. Something really meaningful happened. And he knows that. He still goes back to the same spot. It's never happened again. And that would be the case for us, too. I don't know what your bacon is, but maybe it's a great idea you have, or it could be bacon, it could be chocolate, it could be a first kiss or something. But when those events happen, the idea is that anandamide would get released from the postsynaptic side to go back and say, aha, mark this. This is critical. And that plays a role in forming long-term memories. So that's how anandamide and 2-AG work. But what about THC? Remember, those receptors are everywhere. So when you smoke marijuana, all the receptors get filled, and everything is marked. Everything is salient and interesting and important, which is, frankly, part of the fun, right? So it's, I can remember this, it was with the same friend who I drank all the wine with. We came home one night stoned and found rice-a-roni in the cabinet. And I was like, oh my gosh, this is delicious. How do you keep this on the shelves, And now I wouldn't eat rice-a-roni for anything, but at the time, it doesn't matter. It was so amazing. And the music is amazing. And the texture of the floor is amazing. And this person is amazing. And this idea is amazing. So it's all just totally amazing. And the reason for that is because everything's being highlighted, which is fun. Let's admit it. Amazing and fun. Okay. But there's some consequences of that, beside the fact that everything seems amazing, and that's the way we say that, is that the stimuli are all really salient or really important. We also stimulate pleasure because it releases dopamine in that pathway, along with lots of other things. But because you're flooding the synapses, all with anandamide, you can't learn. Your memory is usually impaired because, especially with an acute dose, you'll see, because part of learning is deciding what to store and what not to store, and everything seems important, so it's going to impair that. It slows your response time, and it makes you have critical tracking errors. And that doesn't sound so terrible, but what I think is problem, and I hear this all the time because I talk to students who say, look, I've been smoking pot for years and I can do just fine in school. I don't have critical tracking errors and I can remember things. I'm doing well in school. How could that be? That's called tolerance, isn't it? So the drug produces less and less of an effect. And how does it do it with THC? It does it, I'm kind of going to show you in a few slides quickly. It does it because this is the number of those receptors, those CB1 receptors, the density of those, in a naive animal and in an animal that's had 4 days of THC injections. So many fewer receptors. And here the same finding is in another study, pictorially. So they had 6 days of escalating doses, not such high doses, kind of recreational doses. And after 6 days, here's the vehicle-treated animals, so it got a placebo, and this is the one that got THC. Many less black spots, right? Here's the last one of these. I just love these pictures. This is what happens when you spend too much time in the lab. But here's an animal that got saline, and this is an animal that got one pretty high dose of a THC analog, a synthetic analog, and you can see it's much lighter. And these animals got repeated dosing for 8 days of a low, medium, or high dose. And you can see in the end, here, 8 days, this is so much less dense than this one. So that's why they're tolerant, that's why they can learn, that's why their critical tracking isn't so bad. It happens in humans too. And here's a recent human study, or pretty recent, where all these yellow areas are where there's significant downregulation of cannabinoid receptors. So these receptors, these CB1 receptors, have to be made about every 3 to 5 days. And when the brain is being flooded with THC a lot, it just stops making the receptors. The longer the human subjects had been smoking, the more downregulation they had. And you're probably wondering, they recover with abstinence. So just like the brain learns to produce this B process to compensate, it also unlearns that, so it can stop that and the receptor levels come back up. So there's really just unequivocal, there are dozens of experiments showing this downregulation, and new research shows that it's stronger in women than in men, and in girls than in boys, for reasons we don't really understand yet. But as a result of that, if you smoke, especially if you're young, your brain structure is altered, and there's lots of evidence for that. The pleasure, the dopamine pathway is also downregulated, which means that, for me anyway, things were really not interesting or pleasurable. The only way I could find something interesting, and I kind of chain smoked marijuana, it wasn't as strong as it is today, but the only way I could find anything interesting was to smoke, because my receptors were absent. It's kind of defeating the purpose. As a result of that, there is an increased risk for other addictions. And this is sometimes called the gateway theory, and some people think, well, and I was one of these actually initially, I thought, well, it's just because if you use as a teenager, you're more likely to develop a problem. Doesn't mean that the early using causes the problem. It could just be that people like me who like drugs like them early and they like them late, and they're likely to try these things. Remember, they like novelty too. So drugs are novel stimuli. But it turns out we've done the experiments now in non-human animals, and just a little bit of exposure during adolescence, because it alters your brain structure and your dopamine pathways, makes you not enjoy drugs as well. And because they're not as enjoyable, you have to take more to get the same effect. You're sort of innately tolerant. You smoke pot, you're innately tolerant to alcohol and to heroin. Therefore, you're more likely to take them because you sort of need to up the ante a little. More likely to commit suicide, more impulsive, and less likely to graduate from high school, and more likely to be depressed. The final sort of bad news about this is that these effects look like they're going to go across generations. So in non-human animals, when we give adolescents exposure to a little bit, 8 days or a week of THC, and then we take it away, we let them grow up, we let them mate, their offspring, their children and their grandchildren are also at increased risk for negative mood and increased risk for taking more drugs of abuse. And that's a whole new area of research. So just to sum up, I told you that I can't wake up without caffeine, and drinking alcohol ends up reducing the amount of pleasure you experience, causing anxiety and causing insomnia. Benzodiazepines that many people take to help them relax and sleep actually produce anxiety and insomnia. Ecstasy or MDMA, something that is pretty widely enjoyed and makes people feel at one and well about things, and it causes depression. So the bottom line is that in every case, the brain adapts so that it produces the exact opposite effect that you're going for. And so for this reason, I'm not so much against drugs, because I think that it's a natural drive to try to change the way we feel. But the B process makes it regular use kind of ineffective, because it diminishes your capacity to enjoy the drug. It produces the opposite effect that you're taking the drug for, and those things are core attributes of addiction. So that is the end, and thank you for your patience.